So today I'm going to be presenting my Genetics is Awesome presentation. So to start, for my late article, I picked an algorithm to detect maternal autoantibody related autism. And how does it work? A computer generated algorithm has been developed to detect the autoantibodies from a mother to determine whether or not her child will subsequently have autism. So the autoantibodies that were used as the markers were the CRIMP1, GBA, and LDH proteins. How it works is these green prongs up here are the maternal autobrain antibodies. They bind to the fetal receptors, which are the purple little prongs down here. All this takes place in the blood plasma of the mother and the fetus. So then by virtue of blood being everywhere in the body, it travels to the brain and then attacks the brain tissue and the antigens in the brain tissue. And this picture down here is just a more simplified version of how that works. So as you can see, it transfers from the mother into the baby's brain. And if a woman had these, she was about 35% more likely to have a child with autism. And this was very promising simply because before then, there was no real way to test for this. And this is very important because parents who are having children would like to anticipate their child having a neurological disorder. So for my primary article, I picked regulation of SPRI3 proteins by X chromosome and a PAR2 linked promoter in an autism susceptibility gene. So overall, the overall question would be, is the SPRI3 protein a marker for autism? And if so, what is the identifying marker as well as the level of expression? So my figure 3A question, which is the figure I chose to represent, was how does the SPRI3 over and under expression affect neurite growth? And for the hypothesis, it was if the SPRI3 protein is overexpressed, neurite growth will be reduced. So for the methodology, the experimenters took 20 superior cervical ganglion cells from 10 P1C57B16 mice and let them sit for 48 hours. After these 48 hours were complete, they took images of the neurites and neurons and measured the length, because that's the question that we're trying to look for. And then a shoal analysis was performed. And then shoal analysis is just a quantitative measure of the length of the neurites and their branching. So here we have figure 3A, and here's the analysis. So the result, as you can see up here, and I'll zoom in, is that the SPRI3 protein's presence dramatically reduced neuronal branching. So you can see down here, this one has less branching. It looks very small as compared to this one up here, which is very spaced out, has a lot of branching and a lot of different connections. This graph right here is just showing the distance from the soma as the independent variable, and then the dependent variable will be the number of intersections. So you can see that the SPRI3 protein's presence dramatically reduces this. Over here in the left-hand corner, we can see that the length is being measured here in micrometers. And to support our hypothesis, the SPRI3 protein is measuring at about 2,000 micrometers, while the control is measuring at about 3,200 micrometers. So it's a very significant difference. And then for branching, significantly less as well, with the control having about 12, and the SPRI3 protein having about 6 or 7. So what are some of the limitations? They only tested this for Caucasian test subjects. And as we know, Caucasian people are not the only people that can suffer from autism. And so therefore, I thought that was a problem and that it should have been a little bit more diverse in the study. And one thing that I would say would be relevant to the regular world or the real world would be that autism is something that affects a lot of people and not much is known about it. And the SPRI3 protein, not much is known about it. So I feel like that would be something that would be worth delving into more and just finding out more about it, because this could really help a lot of parents and bring a lot of closure and could possibly result in more treatment options. So that is my presentation. Thank you. And yeah.